Most people know that Nazi Germany committed atrocities. That is not what this video is about. This video is about something more specific, and in some ways, more disturbing. During the Holocaust, hundreds of doctors, licensed, educated, trained to heal, voluntarily participated in experiments on living human beings. Not under orders, they could not refuse. Voluntarily, many competed for the opportunity. They kept records. They published findings. They presented papers at medical conferences. The entire apparatus of science was applied to human suffering, systematically, methodically, and with complete institutional support. These are the 9 most disturbing experiments conducted in Nazi Germany. And by the end of this video, you will understand something that medical schools still argue about today: whether any knowledge gained from these experiments should ever be used to save lives. Hypothermia experiments. These experiments were designed to find rewarming methods for German pilots who had ditched in the freezing North Sea. The subjects used to find those methods were concentration camp prisoners. Prisoners were forced into tanks of ice water, sometimes for hours, while doctors monitored their vital signs and recorded exactly how long the human body took to shut down. The doctors noted the precise moment each prisoner lost consciousness. They kept recording after that. Subjects were submerged until their core body temperature dropped to fatal levels. Rewarming methods were then tested: boiling water, body heat from other prisoners, lamps. Most subjects died during the cooling phase. Those who survived the ice were frequently killed by the rewarming process itself. Dr. Sigmund Rascher, who ran the experiments, sent personal progress reports to Heinrich Himmler. Himmler requested updates. The data was recorded in meticulous clinical detail: temperature readings, heart rate, time of death, as though the subjects were instruments, not people. Rascher was eventually executed by the SS, not for the experiments, but for falsifying personal records, unrelated to his medical work. The hypothermia data is still cited in cold water survival research today, and medical ethicists still actively debate whether it should be. High-altitude experiments. Conducted at Dachau from 1942, these experiments were designed to understand the physiological limits of the human body at extreme altitude, specifically to help the Luftwaffe develop survival protocols for pilots. A low-pressure chamber was used to simulate altitudes up to 68,000 feet, well beyond what the human body can survive without oxygen. Prisoners were placed inside with no protective equipment. At those altitudes, the human body does not simply pass out. The doctors documented exactly what happens instead. Subjects experienced violent convulsions, burst blood vessels in their eyes and lungs, and died in prolonged agony as the pressure dropped. Autopsies were performed immediately afterward, sometimes while the subject was still alive, to document the physiological effects in real time. Dr. Sigmund Rascher, who also ran these experiments, described in written reports the precise sequence of suffering. An estimated 70 to 80 prisoners died in the chamber. The reports were filed, reviewed, and stored in official Luftwaffe medical archives. The findings were presented as legitimate aviation medicine research at internal military conferences. Post-war aviation medicine developed safe altitude research protocols specifically because of the ethical void these experiments exposed. Infectious disease experiments. Prisoners at Buchenwald, Dachau, and Natzweiler were deliberately infected with tuberculosis, typhus, malaria, and other diseases to test vaccines and treatments intended for German soldiers. The logic was straightforward. German troops were dying from infectious disease on the Eastern Front. Vaccines were needed, urgently. Prisoners provided a controlled population on which to test them rapidly, without the ethical constraints that applied to German subjects. Prisoners were infected through injections, contaminated wounds, and forced exposure. Those who developed the disease were given experimental treatments, some untested, some known to be toxic at certain doses. Control groups were deliberately left untreated to provide comparison data. Mortality rates in the typhus experiments at Buchenwald reached approximately 90% in some test groups. Detailed records of symptoms, disease progression, and time of death were maintained throughout. The doctors running the program had full knowledge of the death rates and continued. Several of the vaccines tested showed no efficacy, meaning prisoners died to confirm what better-designed research could have established without human subjects. The Nuremberg doctors' trial of 1946 resulted in 7 death sentences specifically for infectious disease experimentation. the first time medical personnel were held criminally accountable for research conducted on unwilling subjects. Bone, muscle, and nerve transplants. At Ravensbrück concentration camp, German surgeons removed bones, muscles, and nerve segments from living prisoners and transplanted them into other prisoners, or simply discarded them, to study tissue regeneration and transplant viability. The subjects were primarily Polish women, many of them resistance fighters, who had been selected specifically because they were considered racially and politically expendable. Anesthetic was used inconsistently, in many documented cases, not at all. The women who survived these operations called themselves the rabbits. They testified at Nuremberg. Entire sections of leg bone were removed while subjects were conscious or minimally sedated. Nerve segments were excised and reattached in different configurations to observe regeneration. Some subjects had limbs amputated entirely for transplant studies. Infections were deliberately introduced into surgical wounds afterward to test sulfonamide antibiotics simultaneously. The same prisoners were used for multiple experiments at once. The women who survived were left with permanent disabilities, chronic pain, and in several cases, amputations. They were scheduled for execution before liberation. Witnesses who could testify were considered a liability. Fellow prisoners at Ravensbrück hid the surviving rabbits during final camp inspections, preventing their execution in the last days before liberation. The rabbits' testimony at Nuremberg provided some of the most detailed first-hand evidence of medical experimentation. Their accounts directly shaped the Nuremberg Code's requirement for voluntary informed consent. Josef Mengele's twins experiments. Josef Mengele, the SS physician known as the Angel of Death, personally selected twins from arriving transports at Auschwitz for a genetics research program that had no legitimate scientific basis and no defined endpoint. Mengele had a genuine academic interest in genetics. He had a PhD and had published peer-reviewed research before the war. At Auschwitz, he had an unlimited supply of subjects and no ethical constraints whatsoever. Twins were subjected to eye injections with dye to change iris color, causing infections, blindness, and death. Blood transfusions were performed between twins to observe reactions. Organs were removed for comparative analysis, often without anesthetic. In documented cases, twins were surgically joined, an attempt to create conjoined twins artificially. When one twin died, the other was frequently killed immediately, so simultaneous autopsies could be performed for comparison. Approximately 1,500 pairs of twins passed through Mengele's program. Fewer than 200 individuals survived. Mengele fled to South America after the war and was never captured. Mengele lived freely in Argentina and Brazil until his death by drowning in 1979. Never tried, never extradited, never held accountable for a single experiment. The twins who survived formed an organization called Candles, Children of Auschwitz Nazi Deadly Lab Experiments Survivors, and spent decades ensuring Mengele's crimes were documented precisely because he escaped justice entirely. Seawater experiments. At Dachau, in 1944, approximately 90 Roma prisoners were forced to drink only seawater for up to 12 days to determine survival limits for German pilots and sailors stranded at sea. Two competing methods were being tested, one involving unmodified seawater, one involving seawater treated with a substance called berketite, which disguised the taste but did not remove the salt. Subjects had no access to any other fluid source. Some prisoners were so desperate for water, they licked the floor after it was mopped. This was documented in the experimental records as a behavioral observation. Seawater causes progressive dehydration, even as it is consumed. The salt content forces the kidneys to expel more water than the body takes in. Subjects developed severe kidney damage, seizures, and organ failure. The berketite method proved no more survivable than unmodified seawater. Several subjects died. Others suffered permanent kidney and liver damage. The Roma subjects had been selected specifically. They were considered racially expendable under Nazi ideology, making them the default population for experiments where death was an expected outcome. Their names were not recorded in the official experimental documentation. The experiment concluded that there was no viable method for surviving extended seawater consumption, a conclusion reachable through existing physiological literature without any human subjects. The deliberate exclusion of Roma victims' names from Nazi medical records meant their suffering was systematically erased. a pattern of invisibility that persisted in Holocaust documentation for decades afterward. Sulfonamide drug experiments. At Ravensbrück, wounds were deliberately inflicted on prisoners infected with bacteria, including gas gangrene and tetanus, and then treated with sulfonamide antibiotics to test their effectiveness for battlefield wound treatment. The subjects were primarily the same Polish women used in the bone and nerve experiments. Some were subjected to both programs simultaneously. Wounds were cut into legs, contaminated with bacteria, dirt, and glass fragments to simulate battlefield conditions, then sewn shut to encourage infection. Sulfonamides were then administered at varying doses and intervals to test efficacy. Control groups received no treatment and were observed as the infections progressed. Several subjects died from the induced infections before any treatment was attempted. Others developed sepsis, gangrene, and permanent tissue damage. The experiments produced data on antibiotic dosing that was distributed to German military medical units and used in battlefield treatment protocols. The women used as subjects had no knowledge of what drugs they were being given or why. Several did not survive. The Polish government specifically requested that the doctors responsible for the Ravensbrück experiments be prosecuted at Nuremberg. Several received death sentences. Modern antibiotic research protocols requiring ethical oversight and informed consent were developed in direct response to what was done at Ravensbrück. The women's suffering became the foundation of the rules designed to prevent it happening again. Sterilization experiments. Nazi scientists developed mass sterilization programs aimed at eliminating Jewish, Roma, and other targeted populations without killing them directly, seeking methods that could be applied to millions without individual surgical procedures. The goal was ideological and industrial. Sterilization on a population scale required a method that was fast, cheap, and undetectable by the subject. Three main approaches were tested: radiation, chemical injection, and surgical removal without consent. Dr. Horst Schumann exposed prisoners to high-dose X-ray radiation directed at reproductive organs, causing severe radiation burns, organ damage, and death in many cases. Dr. Karl Klauberg injected caustic substances directly into the uteruses of women at Auschwitz to cause permanent scarring and blockage. Subjects experienced extreme pain, internal bleeding, and long-term organ damage. Klauberg wrote to Himmler that he could sterilize hundreds of women per day using his method. The program was framed not as punishment, but as science, a contribution to the management of population genetics that Himmler considered essential to post-war planning. Klauberg was captured by Soviet forces, tried, and imprisoned, then released in 1955 as part of a prisoner exchange, after which he resumed practicing medicine in West Germany until survivors identified him publicly. The sterilization experiments directly informed the post-war international prohibition on non-consensual medical procedures, written into the Nuremberg Code, and later the Geneva Convention's medical ethics provisions. The hunger experiments. Systematic starvation studies were conducted on concentration camp prisoners to document how long the human body survives without food, on subjects who were already severely malnourished from camp conditions. The research framing was clinical, measuring metabolic responses, organ failure sequences, and survival thresholds. The subjects were people who had already been deliberately underfed for months. The experiment simply formalized and documented what the camp system was already doing. Subjects were provided measured quantities of food below survival threshold, while doctors recorded weight loss, organ function, cognitive deterioration, and time of death. The data was cross-referenced with pre-existing starvation research to build a comprehensive physiological model of human starvation. Subjects who died were autopsied immediately to document organ condition at various stages of starvation. The research was published in internal medical journals and cited in subsequent nutritional science. It represented the complete inversion of medical purpose, using the tools of healing to document and facilitate death with clinical precision. The hunger experiment records were among the most clinically detailed documents recovered from the camps, their precision a direct reflection of how completely normal the work felt to those conducting it. Some findings from Nazi starvation research were later cited in post-war nutritional science, a fact that remains one of the most contested and unresolved ethical questions in the history of medicine. 9 experiments, hundreds of doctors. One thing they all had in common: None of them believed they were doing something wrong. They believed they were doing science. That is not a defense. It is the most important thing to understand about what happened, because it means the problem was never a shortage of intelligence or education. It was a shortage of conscience, and conscience is not something a medical degree provides. Here's the answer to what I promised at the start: whether the knowledge from these experiments should be used to save lives. There is no clean answer. Medical ethicists have debated it for 80 years and still disagree. What they do agree on is this: the moment we stop asking the question is the moment we stop understanding what made these experiments possible in the first place.